Hi, my name is Mike Salazar, and this is my presentation on Chapter 12 of the textbook, Phlebotomy in Sandflies, the Psychodidae. So, as I said before, Chapter 12 dives into the topic of phlebotomy in sandflies, but what exactly are these insects? So, phlebotomy in sandflies refers to this diverse group of species classified under the subfamily Phlebotominae, which belongs to the larger family Psychodidae. And this family encompasses a variety of fly species, many of which we are familiar with as we encounter them in our everyday lives, such as drain flies, sewer flies, sink flies, as well as some species of gnats. So, phlebotomy sandflies are of particular interest to scientific research as they are the sole vectors of leishmaniasis. And leishmaniasis is a parasitic disease caused by a protozoan that significantly impacts public health in tropical and subtropical regions worldwide. This disease can manifest in various forms, ranging from small cutaneous lesions that can form on the skin to life-threatening visceral infections, which makes studying these flies critical for disease prevention and the safety of public health. So, interestingly, the term sandfly is somewhat misleading. While it suggests that these flies have an affinity for very sandy, arid environments, this species often thrives in warm and humid regions, and rather than these arid, sandy landscapes. So this name originates from sandfly species that were originally found in sub-Saharan Africa, as well as the Middle East, where sandflies are common in these regions, though they are not exclusive to them as their habitats. Phlebotomy sandflies also do not exhibit a anthropophilic tendency, meaning they aren't necessarily attracted to humans, although they do serve as vectors for human diseases. And this is mainly because humans often provide areas where these sandflies can oviposit their eggs and their young can grow and prosper. And usually these are regions where there's lots of organic matter for their larvae to feed on. And not all sandflies are actually near humans. There are actually many species of sandflies that live in very remote, uninhabited areas, and so they have this really cool adaptability to occupy several diverse ecological niches. So let's dive more into the taxa of the sandfly. So as we had said before, phlebotomies are part of the Psychodidae family, which has two main subfamilies. And these are the sandflies and the mothflies. And within the sandflies, we have our Phlebotominae. And actually, within the subfamily, you can further go on to organize it into New World and Old World species. So, New World species would be sandfly species that you would find in the Americas, such as in South America as well as Central America. And the Old World species belonging to Africa are commonly found in places like the Middle East, and they're also widespread throughout the continent of Africa. And all of these species can serve as vectors for leishmaniasis. So Lutomia, Brumptomia, Wania, as you can see here in this figure at the top left, these are all common in the New World, and they are all vectors of leishmaniasis. And originally, during the time of writing of the book, Lutomia was regarded as its own species, but has since been reclassified as a complex of sister siblings that are widespread throughout South and Central America. And these species also take on distinct features depending on the environment they occupy, which means that a Lutomia sandfly from the arid mountainous regions of Mexico is going to be slightly different from its tropical counterpart in Brazil. So much of the early classification of sandflies was done through their adult distinctions, because access to eggs and larvae in the field was highly difficult to come by. But since then, genomic sequencing, as well as improved rearing and field methods, has made classifying the sandflies much more accurate and easier, rather than solely relying on these adult characteristics. So right there in the top right corner, you can see an adult Lutomia sandfly. They have this like very almost fuzzy like appearance, and they have a characteristic V wing shape that is prevalent throughout that whole subfamily. And right below that, you can see a actual figure from a research article that where they were able to discover several new species of sandfly within Brazil through genomic sequencing. So let's talk about the actual identification of the phlebotomine sandfly. So as we had stated previously, they belong to the family Psychodidae. So they'll take on features that are characterized within that family. And these include narrow scales that will cover the head, thorax, and wing veins. And these scales are very small and they occur very frequently. So this is what actually gives them that fuzzy-like appearance. And this helps distinguish them from other species of dipteran and other families as well. But whenever you start to focus in on the phlebotominae, there are several key characteristics that set them apart from other members of the Psychodidae family. The most notable part, of course, is their specialized mouthparts. So, phlebotomine sandflies have piercing and sucking mouthparts, which are designed to feed on the blood of their vertebrate hosts. And this is necessary for them so they can consume a blood meal and go on to reproduce. And this feature not only supports the role as a disease vectors, but they also leave behind a very painful, characteristic red bite if you were to ever encounter one. So, in addition to their mouthparts, they also have a smaller, more fragile body structure compared to other members of Psychodidae, which gives them a very delicate-looking appearance. And another key characteristic is their V-shaped wings, as I had said before. And when it comes to classifying species within the subfamily, researchers have relied a lot on their external morphology. And there are over 90 distinct adult characteristics that they actually can use to help identify adult species of sandflies, such as their wing veins, their antennal structures, as well as their genital structures. And while the physiological and morphological traits have been the primary means of classification, the main way that it is done now is through genomic methods, such as DNA sequencing and genetic barcoding, which are far more accurate to actually classifying these sandflies. So now let's talk about the life cycle of the sandfly as well as its vector-parasite interactions. So the sandfly has a pretty typical standard fly life cycle. It has 4 distinct stages, them being the egg, larval, pupa, and adult stages. And this is a holometabolous insect, meaning it goes through a complete metamorphosis, which is typical of many dipterans. And so essentially what will happen is that the female will, of course, take a blood meal, and she will use this to help lay her eggs, and it'll provide the necessary nutrients for egg development as well. And the female typically deposits between 30 to 70 eggs in a suitable environment. Unlike mosquitoes, which also take a blood meal for the reproduction, sandflies don't lay their eggs in standing water, but they do prefer moist, dark habitats that are rich in organic matter. So this is like decaying. So this includes like decaying plant material on a forest floor, leaf litter, animal burrows. Cracks in walls is also a pretty common place where sandflies will oviposit their eggs. And these eggs are very small. They have an oval, dark color. They have an oval shape and dark color. And the location of where they actually choose to lay their eggs much depends on their surrounding environment, mostly on the humidity and prevalence on organic matter. So once they're hatched, these eggs go into their larval stage, where they have 4 instars. And over the span of a few weeks, they'll then be in the larval stage and go into the pupal stage. And after this, they'll spend around 4 to 6 days in the pupal stage, and then they will then emerge as the adult sandfly. And the adult sandfly has a pretty short lifespan. So they have a lot to accomplish within that limited amount of time. So now we'll be getting into a little bit more in depth onto the life cycle of the sandfly, and we'll be starting off with the egg and larval stages. So as I said before, the female will oviposit 30 to 70 eggs on a rough and humid substrate. And interestingly, there are distinct features of the chorion of the eggs that they deposit that can be used for a rough taxonomic identification. And these distinctions are things such as ridges, certain cells, as well as microvilli of the chorion. And so the chorion itself is actually this outer casing of the egg that kind of serves as the armor to help protect the egg. So as a taxonomic identification method, it is not very commonly used, but has recently gained some popularity for a general taxonomic identification of novel species. And the larvae are around 2 millimeters once they hatch out of the egg, and they'll progress in size over the course of 4 instars to around 10 millimeters. And during their egg and their larval stages, they will occupy a highly dense organic matter area. So this can be things such as cages where you keep livestock, or it can be a undisturbed part of a forest floor, which gives them a perfect environment to complete these stages of their life cycle. So here are some electron micrographs of those distinct chorion features that I was talking about in the previous slide. So right here, these are pictures of eggs that were found in Nuevo Leon, Mexico, and they were actually used to help classify the species that these eggs belong to. So right here with Sathromia texana, you can see that the chorion has several ridges on its outer layer, which you can see zoomed in on figure D right here. And right below it, we have Trichopygomia triremula, and these have a sort of volcano pattern to them, where they have these very large bumps, and this can actually be used to help identify the species which these eggs belong to. So, prior to the pupation, the larvae move to a flat and dry vertical surface, and they will remain there for around 4 to 6 days before the adult emerges. And the adults we've dove into pretty in depth. The adults have those characteristic V shapes that I have told you about, and the females are larger than males, and they're around 50% heavier on average too. So now we'll be getting into the vector-parasite interaction. So previously, there was no real good model for phlebotomy and parasite interactions, but now with recent advances, there is a very accurate model of how this disease can be transmitted to hosts. So essentially what happens is that the female sandfly will consume a blood meal from a given host, and they may also ingest an amistagote. So amistagotes are protozoans, and they are harmful intracellular parasites. And once these amistagotes are ingested by the female sandfly, they will be released in the midgut of the sandfly, and right here they will become a procyclic amistagote, which basically means they just take on a slightly more motile form of amistagote. So after this, they go through a simple cellular division, and they become a metacyclic permistagote. And while they're in this stage, they will then migrate to the pharyngeal nerve of the sandfly, which will allow it to be transmitted to an unsuspecting host. And this nerve is crucial for the activation, control, and feeding for the ingestion of a blood meal. So that is actually how that amistagote is able to migrate from the midgut of the sandfly all the way to the mouth part for it to be transmitted. So once the sandfly has that amistagote in the pharyngeal nerve, it can then bite on to a host. And when it binds onto the host, that promistagote will then invade the macrophages and granulocytes of that host. And during this stage, they will turn back into promistagotes, and then go on to become amistagotes again. And these amoebas can also continuously infect macrophages to keep renewing themselves within that vertebrate host, and then continuing on that cycle where the female sandfly will take a blood meal and then go on to infect another host. So there wasn't a really good model for this before, simply because there was a lot of difficulty in the rearing conditions of the sandflies. It is very hard to grow them and contain them, and although they are mainly researched for leishmaniasis, not all phlebotomy sandflies transmit the same leishmania species. So this is because leishmaniasis is just this broad term for a disease that is caused by the Leishmania protozoan, which can take on many distinct characteristics depending on the environment as well as geographical location. So let's get more into the sandfly habitat and their environmental interactions. So in general, phlebotomies are categorized as peridomestic or feral. Peridomestic meaning they live close to human activity, or feral meaning that they live in remote wilderness areas within their geographical constraint. So, as I said before, sandflies will likely inhabit anywhere where there's a abundance of rich organic matter, there's lots of moisture, there's a crevice to hide in to protect their eggs, and so there's a lot of places within their environment where they will lay their eggs, such as within animal burrows, caves, the husks of trees, a bedding of rocks, anywhere that kind of provides this texturized environment where they're going to have a lot of humidity and easy access to nutrients is a strong environment for the sandfly. So these sandflies aren't just in the wilderness, they are very close to humans as well, because we provide those environments for them. So one example of this is this coconut shells right here. So sandflies like to lay their eggs anywhere where there is decaying matter, and these coconut shells are a perfect opportunity for a sandfly to go in and oviposit their eggs and provide them a lot of nutrients for their young, and so on. So this also includes domestic animal enclosures, which is where the majority of leishmaniasis infections take place, where they're very close in proximity to animals, their waste, and so on. And these sandflies have an average flight range of less than 60 meters, so they can't really fly too far from where they are oviposited, and they'll generally stay within that nearby vicinity. And forest species are much less active, meaning they don't travel nearly as far as their peridomestic counterparts. So now that we've taken a pretty detailed look at the environment and habitat of sandflies, let's take a broader look and look at their global distribution. So, as I said before, these tropical, subtropical regions are excellent environments for sandflies to thrive, but temperate regions also provide excellent environments as well, such as some areas of the Mediterranean and some areas of the United States as well. In the Old World, the genera Phlebotomus commonly dominates and is widespread throughout the continent of Africa, and in the New World, Lutomia is the prevalent genera, and it is found all throughout South and Central America. And these species are highly adaptable, but they do not do well in areas with high altitudes or polar regions. This is because they are weak flyers, and any small change in the air can throw them off course and potentially harm them. So they typically stay within their respective regions where it's very low, there's not much air, and very humid. As well as some changes in climate have helped expand the areas in which sandflies are prevalent. So the global increase in temperature has also made some areas in the United States much more viable for sandflies to live. One of them being Texas, where we have seen an influx of sandflies, and we have also have our own native species of sandflies as well. So now that we've learned more about the biology of the sandfly, their geographic location, their habitat, and their environment, let's talk about why this is important to us. So, as I stated in the beginning, sandflies are the sole vectors of leishmaniasis, which is a terrible parasitic disease. So, leishmaniasis can manifest in two main forms, this being cutaneous and mucocutaneous leishmaniasis. So, the cutaneous form is much more common, and it causes skin sores that will then develop into bumps or nodules that then develop into these very painful ulcers, which leave lesions as well as significant scarring as well as disfigurement. Though this form is usually non-life threatening, its counterpart, mucocutaneous leishmaniasis, is much more severe. As the name suggests, it affects the mucous membranes of the nose, mouth, and throat, leading to significant tissue degradation, breathing difficulties, as well as facial disfigurement. And this form often goes untreated, as it takes years or months to develop after the initial skin infection has healed. So, a very fatal form of leishmaniasis. Treatment for the disease typically involves antiparasitic drugs like amphotericin B or methotrexate, but access to these treatments can be challenging, especially in the rural and impoverished areas where leishmaniasis is very prevalent. And without the proper medical care, both of these forms can lead to severe complications. However, beyond leishmaniasis, these sandflies can also transmit other diseases. So one example is Bartonella bacilliformis, which is transmitted by sandflies in Ecuador and Peru, which causes carrion disease. And if you're not familiar with it, it is a very life-threatening bacterial disease which causes anemia as well as severe skin lesions, similar to leishmaniasis. Other diseases include vesicular stomatitis virus, or VSV, and this disease has a more agricultural impact as it more commonly affects livestock and occasionally infects humans. And it is commonly associated with the Lutomia shannoni species of sandfly, which is also very prevalent within South and Central America. And in summary, while these sandflies are small, their impact on human health and animal health is very significant, and it establishes their role as a vector of awful diseases and highlights their importance for a public health effort to control the populations, improve disease surveillance, and as well as expand healthcare in these vulnerable regions where leishmaniasis is very prevalent.